Hello again, everybody. Welcome to the webinar on Steps for Success with RNAscope Multiplex Fluorescent Assay. My name is Sudha Krishnan, Technical Support Specialist, and I'll be discussing some key pointers and suggestions for a successful multiplex fluorescent assay. A few points about the webinar. The audience will automatically be muted. If you would like to ask a question, we request that please select the chat button to type in your questions. If we are unable to answer your questions, we'll follow up with you offline. For today's webinar, we'll start off with a brief introduction to RNAscope in general. Within this segment, I will also be covering the multiplex fluorescent workflow and follow up with some tips and suggestions for the fluorescent assay. I will then wrap up with some troubleshooting tips and provide time for Q&A. If time permits, I will also take you through a test drive of our acdbio.com website, wherein I will show you where to obtain the user manuals, how to get more information on our products, and so on. So RNAscope is based on proprietary in situ hybridization technology, which is based on unique probe design and signal amplification system. Now, just as in any in situ assay, RNAscope assay involves permeabilizing properly prepared samples to allow for complementary oligo probes to bind to the RNA of interest. Now, specific signal is then picked up by the double Z probe system and amplified by a series of amplification cascades. Now, the signal, which is subsequently detected, is visualized as punctate dots and can be quantified. Now, as in the case with the multiplex fluorescent assay, you would use a fluorescent microscope for this purpose. Now, thus, through a unique combination of our probe design and proprietary signal amplification system, we are able to present you with an assay that is extremely universal, as well as rapid and scalable, with the capability of single molecule detection in single cells for virtually any genome, any gene, any tissue. So how does this technology work? Now, RNAscope, as I mentioned before, the RNAscope probe design is the key. We use double Z probe design, and there are three components to this Z. You have the lower region right there, which is the 14 base tail sequence, and is the region where, we have the lower end region of the Z, which then is right there, which binds directly to your mRNA. Now, this lower region contains 8 to 25 base sequences that is specific to your target of interest. The upper region right there is a 14 base tail sequence and is the region where your preamplifier binds and the cascade amplification occurs. Now, we also have a spacer in between the lower and the upper, and that allows for flexibility. As our users are aware, these Zs are used in pairs. In pairs, the upper region, right there, the upper region of the Zs will provide 28 bases for preamplifier binding. Now, both these Zs bind adjacent to each other to your target region from the lower region. Now, why is it that we have the double Z probe system? Why is it that we have the Zs in pairs? Now, if a single Z binds to a non-specific RNA, the preamplifiers will not be stable on just 14 bases. So the non-specific binding that may occur is actually not picked up and amplified. So this is how the double Z design prevents background noise and contributes to specificity. There is one more reason to it. It offers simplicity of design. Why is that helpful? As you can see here, let's say the yellow line, that represents your RNA of interest. If your RNA is intact, your assay will hybridize up to 20 Z probe pairs to your RNA. But then in many cases, let's say you're working with cancer FFPE samples, let's say you're working with archival clinical samples, your RNA could be degraded and fragmented. The simplicity of this design with relatively short probe binding to your target allows for binding to even short pieces of RNA. Thus, what we present here is a highly specific and robust assay. Now, let me delve into the RNAscope workflow. Before I go into the actual workflow, let me introduce some of the details about our multiplex fluorescent assay. Now, with this assay, we have the capability of detecting up to 3 targets at the same time. The fluorophores come in wavelengths that are similar to FITC, Psi3, and Psi5. The probes for this assay would come in channels 1, 2, and 3, which are annotated as channels C1, C2, and C3. In the next few slides, I will go over in detail about how to combine these probes, and I will also be addressing some questions regarding the channels. What do these channels, the C1, C2, and C3 actually mean? Here is the workflow for the multiplex fluorescent assay. As you can see, it has 4 amplification steps, and the signal is detected using a labeled probe that is tethered to a fluorophore. The signal can be detected in up to 3 microscope channels. We have the Alexa 488, Auto 550, and the Auto 647 channels. I would like to take a minute here by talking about the probes, about how to plex our probes. So basically, for the multiplex fluorescent assay, we offer probes in 3 channels: C1, C2, and C3. This allows for detection of up to 3 targets at the same time. So in a scenario where our customer is using that scenario in which we are detecting 3 probes, 3 targets at the same time, you would have a C1 probe, which is ready to use, which is provided at 1x concentration, and you would have a C2 and C3 probes, which are actually concentrated, which are provided at 50x. So what the customer would do is reconstitute or combine the C2 and C3 in C1 in the ratio of C1 is to C2 is to C3, 50 is to 1 is to 1. That's how you would make a 3-plex probe mixture. Now, let's say your experimental design requires you to visualize C2 alone, or C3 alone, or C2 and C3 alone. We have the option for that as well. We do provide a blank probe C1, which is just a probe diluent. So you would prepare your multiplex or your probe mixture with C2 and the probe diluent in the similar ratio. Now, here is the workflow in detail. As you can see here, what we show is the workflow, which involves pretreatment, hybridization, amplification, and so on for fresh frozen sample type. That is because the sample type which is recommended for our multiplex fluorescent assay is fresh frozen. We do understand that our customers have various types of sample. They might be working with FFPE and fixed frozen. If you have any questions on how to perform your multiplex fluorescent assay on those sample types, we would be very happy to speak with you. So as I mentioned before, this workflow is for the fresh frozen samples. So the pretreatment simply involves protease digestion. Some of our customers, again, who are used to working with FFPE and fixed frozen samples are used to a heat-mediated target retrieval step, which is not required for fresh frozen samples. So all the pretreatment entails, in this case, is fixation, a very, very brief 15-minute fixation at 4 degrees in PFA, followed by dehydration and protease digestion. You would then move on to target probe hybridization, washes, pre-amplifier binding, and amplifier binding. You would then follow up with the labeled probe hybridization, some washes, DAPI counterstaining, and followed by image detection under the fluorescent microscope. Now, one suggestion or one thing that I would like to mention at this point is that pre-treatment conditions will vary based on sample type. So there are two ways to determine this. One is you may download the appropriate user manuals, and towards the end of the webinar, I will be showing how to download the relevant user manuals from our website. The other suggestion I have is that we have a very detailed matrix under our products page, which gives details on which pre-treatment to use for which sample type. Now, our customers' convenience and flexibility is very important to us. So we also recognize that our day-long assay can be a bit long and intense. So what we have done is we've developed this assay such that our customers can do it in one whole day, or you can split it up into 2 days. So if you do follow the one-day assay format, you will do everything starting from sample preparation to sample pretreatment onto RNAscope assay in 1 day. If you would prefer the 2-day format, then we recommend that our customers do stop at sample pretreatment on day 1 and then start up with the RNAscope assay the next. In addition to that, I would request our customers to please refer to our Part 1 and Part 2 user manuals, where we have multiple optional stopping points. Now let me switch gears a little bit and talk about some tips for the RNAscope manual assay. In a very, very broad, high level, here are the 4 tips. The first one would be to follow the protocols exactly as described in the user manuals. Now, this is because our ACD scientists have done a lot of testing, and the user manuals are highly detailed, and they are, again, compartmentalized based on the sample type that our customers might have. So I would request that you please follow the protocols as described in the user manuals. The other tip would be to review the pretreatment conditions or recommendations that are specific to your sample type. As I mentioned, we support the assay on FFPE, fixed frozen, fresh frozen, as well as cultured adherent cells. And each of these sample types have different pretreatment recommendations. So I request our customers to please review sample pretreatment recommendations for their particular sample type. The next tip would be to always use controlled probes and slides. I'm going to be going over this part in detail in the upcoming slides. And finally, last but not the least, is review that you have, you're using all the required materials. And before you start your assay, make sure you have all the critical must-haves for the assay. If you have any doubts or any questions regarding this, please do get in touch with us, and we'll be more than happy to go over your sample type and the assay that you're running and provide all the critical must-haves for your assay. This is what the checklist, a generic checklist for the multiplex fluorescent assay would look like. The very first requirement is the image hydrophobic barrier pen. Now, this is the pen of choice, the hydrophobic pen of choice for our assay. It has been extensively tested and validated by our ACD scientists to hold up a very strong hydrophobic barrier around the samples, and this prevents leakage and drying. The next one is positive and negative control probes that are species specific. Hot plate for pretreatment or the target retrieval step. This is required for FFPE and fixed frozen samples. We also have a protocol for steamers if our customers would prefer using a steamer for this particular step. The only take-home message here is that this target retrieval is to be done either in a hot plate or a steamer. We do not recommend the use of microwaves or a pressure cooker. The other critical requirement is SuperFrost Plus slides. Now, that is because our assay has multiple hybridizations and washing steps, and we do not want our customers to lose their samples halfway through the assay. And upon extensive testing, it has been determined that this is the slide that promotes proper attachment of the samples throughout the assay. The other critical requirement is the HYBEZ hybridization system, and I will be talking about this in greater detail in the upcoming slide. The other requirement would be RNAscope control slides, and I'll be touching upon this in the upcoming slides as well. The mounting media of choice for the fluorescent assay is prolonged gold mounting media, and we also recommend that our customers use fresh reagents for our assay. Now, this is a typical setup, hot plate setup for target retrieval. The most critical points to watch out for are the temperature of the buffer and duration of boiling. Now, if either of these parameters are overextended, the tissue will be overdigested. Now, one suggestion we have for this step would be to make sure that the buffer is kind of at a gentle boil. And what I mean by that is that you should watch out for, very fast jets of bubbles shooting up from the bottom of the beaker. That's what we watch out for that, and that is, we do not require, that would be harmful for the, if the bubbles are shooting up from the base of the beaker, samples might get detached. So if you do observe this, we suggest that our customers try to position the slide rack away from these bubbles, reduce the temperature a little bit, and work with it. One other recommendation is to cover the beaker with a foil to prevent boiling over. Now, this is what a typical RNAscope reagent kit would look like. The RNAscope reagent kit comes with pre-treatment reagents, detection reagents, and wash buffer. And we do recommend that the probes be warmed at 40 degrees for 10 minutes before use. And the wash buffer comes as a 50x, while the target retrieval comes as a 10x concentrate. Please do warm it at 40 degrees for about 20 minutes before reconstituting. Now, shown here is our HYBEZ hybridization oven. Now, customers who have been in touch with me have heard me talk about this oven very often. It has been consistently validated by our scientists to be the very best for our assay. Now, this is because it has been engineered with our assay in mind and shown to maintain the perfect temperature and humidity environment for RNAscope assay. The oven comes with a humidity tray, a slide holder, and humidity sheet. Now, it is critical to moisten the sheet and place the humidity tray in the oven for some time before running the assay. Now, what I would like to mention at this point is that I can assure you that if you're using this oven on properly prepared samples, your assay will be a success. So if you have any questions on how to acquire this oven for your needs, I would urge you to please contact your area accounts executive. Now, shown here are some the accessories for washing steps. So on the left, our customers might be using Tissue Tech washing tray, which is also compatible with RNAscope Assay. And shown here is the Easy Batch slide processing system. Now, let me take a minute describing the system as it is a very, very nifty tool for streamlining your RNAscope assay. It comes with a slide holder, as shown here in off-white, as well as a wash tray. Now, this slide holder, as you can see here, the slides can actually fit in the grooves right here, and you would simply pinch these these two ends, and you would lock the slides in place. When this is done, you can seamlessly go between the RNAscope, the HYBEZ oven and washes. So seamlessly go between the amplification steps and washes. Now that's because the slide holder fits in the humidity tray as well of the HYBEZ oven as well. Again, if you feel that the AZ batch slide processing system is something that might work for your experimental needs, please do contact your area accounts executive. Now, here are some guidelines to follow, some generic guidelines to follow for our RNAscope assay. These would entail applying all the amplification steps in the order as described in the user manual. Use the flicking or the tapping technique to remove any residual reagent. Now, in this, when I'm talking about this, let me go back to our easy batch tray. That is because this slide system, this slide holder, it's very convenient if you're using the slide holder to actually remove residual reagent. What you would do is simply hold the slide holder and flick it smartly a couple of times, and all the residual reagent gets removed. These slides are all locked in place, so you do not have to worry about the slides falling off. The other guideline would be not to let the slides dry out. Make sure that the hydrophobic barrier remains intact. And we also request our customers not to alter the protocol in any way. If you think that you have some specific requirements for your experimental need, please do give us a call or send us an email, and we will work with you to understand your expectations and your experimental needs and see how our RNAscope can work for you. The other important guidelines would be to warm the probes and wash buffer at 40 degrees, as they may have precipitated during shipping and handling. Maintain adequate humidity in the humidity control chamber, and again, use fresh reagents. Now, let me dive into the RNAscope Multiplex Fluorescent Assay. Now, let me start off with introducing our color modules, the options for different color combinations that our customers can use. Now, at ACD, we would like to give our customers the option of flexibility in terms of channel choice. Thus, keeping that in mind, our fluorescent assay comes with 3 AMP4 alternatives. Now, these are called AMP4 FLs, A, B, and C. These were allowed for flexibility of switching channels for a target using the same probes without the need to reorder the probes. If you have any questions, if you are looking for more details on this, I would refer you to pages 7 and 8 of the Fluorescent Detection User Manual for more details. However, let me also walk you through these alternatives here. Now, let me give you a hypothetical example and say that we have 3 targets. Let's say we have target X, Y, and Z. Target X is in the C1 channel, target Y is in the C2 channel, and target Z is in the C3 channel. And let's say you use AMP4 Alt-A for the detection. If you use Alt-A, then X can be visualized in Alexa 488, Y will be visualized in Auto 550, and Z will be visualized in Auto 647. If you use Alt-B, then these are your options for visualization. So as I talk about channels, this is a point where I would like to point out the terminologies. So we do understand that our customers might have questions about these channels. So what we have is we have probe channels. Now, probe channels are the C1, C2, and C3, which are actually the tail sequences, which are based on the tail sequences of the probes. We also have, when we talk about the other channel, we talk about the microscope setting channel, which is the Alexa 488, Auto 550, and the Auto 647 channels. Again, if you have any questions regarding this or how best to use or which alt to use for your particular assay, we will be more than happy to work with you. So we have updated our website in which we have more information on our multiplex fluorescent assay, And this is a schematic that can be found on the website as well. So this just reiterates what I briefly touched upon in terms of detection of multiple targets or up to 3 targets at the same time. So again, here, this schematic is a very good visual representation of how our multiplex fluorescent assay works. As you can see, we have, again, 3 targets. So here we have targets 1, 2, and 3 in these 3 channels. And this is how you will be able to visualize these targets at the same time. Now, one of the major steps for a successful RNAscope multiplex fluorescent assay is qualification of the samples, this is the very first step that you would do in your assay. So basically, this is a schematic right here that shows in a very good step-by-step fashion on how you would go about qualifying your samples. Now, this involves actually 3 easy steps. Some of our users may be familiar with the use of the control slides that we offer. We offer HeLa for human and 3T3 for mouse. These are FFPE slides, so many of you who are working with fresh frozen may not be using this step at all, and you would actually be starting from here, which is performing the positive and negative control QC on your target sample. However, let me address the use of the control slides as well. The use of control slides is basically a technique check or a workflow check to make sure that your RNAscope workflow is working fine, the reagents are fine, and so on. So upon successful QC check of this, you would then move on to the sample check. And this is where many of our customers would actually start their assay. What you would do is run negative control probe, which is DAP-B. And what we offer DAP-B as a singleplex and as well as a 3-plex DAP-B, in which we have the same gene DAP-B in the 3 channels that we talked about. So DAP-B, C1, C2, and C3 channels. We also have a positive control probe. Again, we have a singleplex positive as well as a 3-plex positive control probe in which we have 3 housekeeping genes all combined as one probe mixture. So you would hybridize the negative and the positive control probe on the samples and perform 2 things. What you're doing at this moment is checking the RNA integrity of your sample. So based on the number of dots, the intensity, and so on. The other thing is you're looking for background, making sure that your negative control probe is yielding clean background. And you're also assessing the pretreatment efficiency. So at this point, what you'll be looking at is intact morphology. What we don't want is over-digestion of the sample. We also do not want under-penetration of our samples. So having passed all this, so the 3 criteria would be a clean background, intact morphology, as well as expected signal intensity. You would then move on, so that in that case your sample has passed, you would then move on to running your target probe. If for some reason it fails, then here are your options. You can verify, what you can do is you can verify your technique. You can check RNA quality. You can run it using new samples, and you can also perform assay optimization. Now, this right here is just a reiteration of what I just mentioned before, which is your sample qualification, which is it is a visual flow chart. In general, some of our customers may start with step 1, which is using our control cell pellets. If not, our customers may also start here, which is a sample check, running positive and negative control probes on the samples. And if it passes, you would move on to your running target probe. If it fails, you would move on to optimization of the sample, of the assay. So how would you optimize the assay? And why do we even need to optimize the RNAscope Multiplex Fluorescent Assay? Now, we recognize that our customers have many different types of, if they work with different fixatives. The protocols might vary. We have the duration of fixation, the temperature of fixation varies, and so on. But what we have done at ACD is that we have standardized our fixation, and we have determined the best fixation technique that works optimally for our assay. So based on these guidelines, some of the samples that our customers might be working with may be considered underfixed. So they may be considered underfixed if, let's say, they're using 4% PFA for just about 4 to 6 hours. The samples have been fixed just for about 4 to 6 hours at 4 degrees. Or if the sample has not been post-fixed at all. These samples might be considered overfixed when you're using, say, 10% NBF or 4% PFA for greater than 48 hours at either 4 degrees or room temperature. There are also cases in which our customers work with special sample types. They might be working with xenografts, cultured cells, cell pellets, and so on. And in addition to that, we recognize that not all tissues are the same. Some are really tough to penetrate, while some are very delicate. So we have it runs the whole spectrum in terms of ability to penetrate. So we have special tissues, example, liver, muscle, retina, and lymphoid tissue. So these are just some of the factors that actually also affect RNAscope assay performance. The top two would be fixation conditions not optimal and RNA being degraded. Actually, these two conditions go hand in hand. And if this is what you observe, then these are our recommendations. We recommend that you fix the samples as recommended by ACD and or acquire new samples and assess RNA quality. If the hybridization conditions are not optimal, one of the most straightforward ways of troubleshooting this would be the use of the HYBEZ hybridization oven. If you notice samples drying out, again, the most sure-shot way of fixing that would be the use of an image hydrophobic pen. This is speaking to the fact that some tissues are more sensitive than others. If that's the case, then what we recommend is that customers always start with standard pretreatment conditions. And then we would be more than happy to review images with you, and we can provide pretreatment optimization recommendations. So always start with standard pretreatment and then optimize accordingly. So here is how you would perform sample optimization in 3 easy steps. Again, as I mentioned, always start with standard conditions. And what you would observe, let's say you observe a very high background, perhaps your sample's overdigested. That being the case, in general, we have seen that underfixation of samples makes the sample vulnerable to overdigestion. Perhaps you are observing no signal or weak signal. Now, that could be related to the fact that the sample is underdigested, and that might be stemming from the fact that it was overfixed. Now, the ways to address those would be to adjust the boiling time, which is a target retrieval step. The standard for fixed frozen samples is 5 minutes at 95 degrees. You can also work by adjusting the protease time. And here I would like to mention that the standard for fresh frozen samples is 30 minutes at room temperature. Now, one other point that I would like to mention for our customers who are using cultured adherent cells, the protease is to be diluted at 1:15 in 1x PBS. Now, here is what a successful RNAscope multiplex assay would look like. What I'm showing here is the use of a 3-plex positive control, and here we have used Alt-A to visualize these. So thus we are able to see polar 2A in the Alexa 488 channel, PPIB in the Auto 550 channel, and UBC in the Auto 647 channel. You may notice that the expression patterns are different. So this is the reason, this was the rationale behind the choice of this combination for positive controls. Polar 2A runs, in general, it's a low to medium expressor. PPIB is a medium to high expressor, while UBC is a high expressor. Now, let me talk about some troubleshooting tips for the multiplex assay. A very commonly observed issue that we have come across is sample overdigestion. Basically, again, the example that I'm showing here is for a fresh frozen sample, and the recommendation is a protease digestion at room temperature. Sometimes if this particular step is done at 40 degrees, the sample gets overdigested, and here is what I'm showing as an example of an overdigested sample, and the best way to fix it is running the protease digestion at room temperature. Now, sample thickness. The recommended guideline or the recommended range for fresh frozen samples is 10 to 20 microns. Again, this is based on our testing. What we're looking for is the thickness that promotes best sample retention as well as provides good signal intensity. So that's, as you can see here, 10 to 20 microns is a range that we recommend for fresh frozen samples. I would like to take a moment addressing autofluorescence. Now, some of our users who are using fixed samples, it could be fixed frozen, it could be FFPE samples, may run into the phenomenon of tissue autofluorescence. Now, that is because the fixation process introduces crosslinks, and that causes, that leads to this process of the phenomenon of autofluorescence. One way of correcting this is the use of a multispectral imaging system to correct for or reduce autofluorescence. In-house, we use a software called Nuance. This is from PerkinElmer. So as you can see here, I'm showing some examples of FFPE kidney, intestine, colon, and brain, and it shows varying amounts of autofluorescence. I would like to wrap up this section by just bringing up some of the commonly observed issues with some troubleshooting issues that we have seen with the multiplex fluorescent assay. One of the major points would be microscopy. Now, let's say we see a no signal, weak signal, non-specific signal. Now, these are perhaps related to the use of wrong, let's say wrong filter setting in the microscope, or longer emission cutoff. It could be related to wrong exposure, or the inappropriate or usage of an imaging system which is not probably compatible. So the solutions for these would be correcting the filter settings, and also we recommend not using the auto exposure at first. Please do verify signal with naked eye, and again use the Nuance, use an image enhancing software like Nuance. Now, at this point, I would like to point out that use of a 3-plex positive control can help in assessing your microscope settings. The other issue that we have run into with multiplex fluorescent assay could be the sample. Let's say our customers are seeing no signal, weak signal. Again, that's perhaps related to compromised RNA quality or sample preparation, like say high autofluorescence or background in the sample. To fix this, we recommend using a new sample with good RNA quality. Please do follow the pre-treatment guidelines as recommended. And again, always perform the assay with a 3-plex positive and 3-plex negative control probe. This not only helps you qualify your sample, it also helps you assess your microscope settings. And again, always check the signal with naked eye under the objective lens first. Now, I would like to close this session off by bringing up some of our frequently asked questions. So these would be, let's say, what is the assay compatibility of RNAscope? As I mentioned, our assay works with FFPE, fresh frozen, fixed frozen, and cultured cells. However, for the multiplex fluorescent assay, we do recommend that our customers use fresh frozen samples, as that is the sample type of choice. Some key differences between RNAscope assay and IHC for, say, FFPE samples. No cooling is required during epitope retrieval, and users should directly put the slides in water at room temperature, dehydrate, and proceed to protease digestion as per Part 1 of the manual. Now, at this point, what I would like to do is take our customers through a very brief test drive of our website. I would like to show our customers where to obtain our user manuals. So please do go to, when you go to acdbio.com, I would recommend our customers please visit our support webpage where there's a wealth of information. And in order to get download the user manuals, please select the option for user manuals. Upon doing so, you will be presented with this interactive guide. You would choose the platform, say manual platform, and fresh frozen samples. And let's say we are doing the multiplex fluorescent assay. When you hit search, you will be presented with some documents. And I would like to draw your attention to the top two. The first one is part one, which is sample prep. It gives details on how to prepare your samples as well as specific pretreatment recommendations. And part two would be the actual detection or detection workflow. Under the support page, we also have wealth of information in terms of FAQs, troubleshooting guides, and so on. Now let me draw your attention to our pretreatment matrix, which I was talking about. So if you go to our products page and select manual assays, you would then select RNAscope pretreatment, and you scroll down, and we have this very detailed matrix, which provides a very good snapshot of what type of pretreatment would you require. So let's say you are running fresh frozen multiplex fluorescent, then the only pretreatment that you would require would be protease 4. On the other hand, let's say you are running fixed frozen, you're running the multiplex fluorescent on fixed frozen, then you would have to perform target retrieval followed by protease 3 digestion. And as I mentioned before, we have revamped our product page for the fluorescent assay. And I would like you to, I would like to request our customers to take a look at our fluorescent assay webpage, where there is detailed information on our technology, as well as information on how to get started and some guidelines for channel selection and so on. At this point, I would like to address some questions that our users have put to us. Again, the one question is, what is the difference between a probe channel and a fluorescent filter channel? So a probe channel is it's annotated as C1, C2, and C3. It is different from the fluorescent channel or the microscope, or the fluorescent filter channel, that is FIT-C, Psi3, and Psi5. So a probe channel actually refers to the signal amplification and detection system to be used for a particular probe, which is again designated as C1, C2, and C3. The ability to assign different target probes to different signal amplifiers and detection systems is the basis for our multiplexing design. One more question that we have is, let's say a customer doesn't know how the tissue was prepared. So I'm not sure how the tissues were prepared. What pretreatment conditions should I use? Now, basically, what we would recommend is always start with the standard pretreatment conditions, but you would refer to our pretreatment matrix to actually use the sample type specific, assay specific pretreatment. Now, what controls do I need for the multiplex fluorescent assay? We recommend that our users use species-specific positive control probes for samples. So we have, say, if you're working on mouse brain, you would use mouse-specific controls, and a negative control, which is the non-specific bacterial gene. As I mentioned before, we recommend that you use only a 3-plex positive and 3-plex negative control probe. Now, the reason again for these are you'll be able to see signal in 3 different fluorescent filter channels. This is a technique check, it's an RNA quality check, it serves as a microscope setting check, and also you are able to assess background signal in all 3 channels. The other question that we have that just came in would be, why do I need a hydrophobic barrier pen for the fluorescent assay? This is because you would need it to create a strong hydrophobic barrier around the tissue when you perform the assay. Now, again, this is the image pen is the only pen that we have validated that creates a hydrophobic barrier that is maintained and does not leak throughout the entire RNAscope protocol. Now, if leaking does happen, it will cause the samples to dry out and in many cases contribute to autofluorescence. The other question that we have is, do you have a 2-plex kit designed for the fluorescent detection of RNA in my tissue? Now, basically, for the fluorescent assay, you can visualize 1, 2, and up to 3 targets. So thus, the answer is yes, you can use probes in 2 different channels in a 2-plex situation to visualize only 2 targets. As far as other things goes, nothing changes with respect to the reagent kit or the pretreatment conditions. Only the probes that you would require for visualizing your target would be different. At this point, I would request our attendees to provide me with questions. If we are able to answer you, if we are able to handle them here, we'll be more than happy to provide you our suggestions. If not, we will follow up with you offline. So we have a question about the preparation and the FFPE liver tissue preparation. So in this case, we request our customers to follow the Part 1 user manual for FFPE samples, specifically for the fluorescent assay. So the sample preparation remains the same, which would be for FFPE, it is 10% NBF, fixed for 16 to 32 hours at room temperature. As far as pre-treatment conditions go, I would like to point out that here for our multiplex fluorescent, for the FFPE, our customers would start with target retrieval, which is the boiling step, followed by protease digestion. We are addressing our customers' questions by the chat, through the chat feature. So if you have any questions, we request you to let us know via the chat feature. We are addressing several questions via the chat feature. We have a couple more minutes left in this session. So if we are not able to address your questions in this session, we will be more than happy to follow up with you offline. Again, thank you very much for your time, and thank you for attending this webinar.